Imagine you're a farmer. You've seen your apple tree sprout from a seed to delicate white blossoms and now juicy red fruits. All of a sudden, you see brown spots spreading from leaf to leaf, then plant to plant, the result of an invisible plague. The plant has been infected by Erwinia amylivora, causing a disease called fire blight, which threatens your crop. It's just one of many bacteria that can infect and kill plants. Like many commercial farmers, you turn to chemical sprays with antibacterial action, but understandably, your customers are worried. Antibiotic resistance is on the rise for one, and the bioaccumulation of chemicals in the wider ecosystem is also a concern. You may suggest genetically modifying your plants, but they're equally worried about this. What are the long-term effects on the body? They want a greener, safer future. What if there was an alternative? Something already prevalent in nature, bacteria's ancient nemesis: viruses. Bacteriophages, or more simply, phages, are viruses that attack and often kill bacteria. They're incredibly specific. Think of the phage like a molecular key. The bacterial surface has molecular structures that can act like locks. If the surfaces interact correctly, the phage would attach. Otherwise, nothing happens. Once attached, the phage injects its genetic material into the bacterium, turning its own machinery against it. It's like taking over a factory and replacing its instructions. The bacterial ribosomes begin producing viral proteins which are assembled into new phages. Soon the bacteria bursts, thereby killing it and releasing all the newly formed phages that go on to attack other bacteria. So, why does this make for a greener future? Well, firstly, phages are entirely natural. You could find them anywhere, from your back garden to the middle of the ocean, and they're essentially harmless to humans and other animals. This means far less agricultural runoff of synthetics that could pollute ecosystems. Secondly, they're ultra-specific, unlike the antibiotics that some currently use. Antibiotics target a similar range of bacteria. For example, penicillin would target a range of gram-positive bacteria due to similarities in their cell walls. This means that while they can kill the target bacterium, they can also wipe out a range of other microorganisms at the same time that may be helpful to the plant, like nitrogen-fixing bacteria. On top of this, exposing the non-target bacteria to antibiotics creates a selection pressure, encouraging resistance to develop. Phages prevent these issues by having to recognise the correct bacterium before even acting. Back to our farmer, him and his customers are much happier with this idea. Now, there are a few issues. Firstly, the specificity means we need to find the right phage for the right bacterium. That's where phage libraries around the world come in, where scientists collect phages, deduce their target bacteria, and then store them for later use. Secondly, the phages are still a selection pressure just like antibiotics. This means that the bacteria with adaptations to evade phages will survive and reproduce, spreading the genetic mutation that caused the adaptation and thus generating resistance. The solution? Phage cocktails. These are mixes of phages that attack one bacterium via different strategies. If one phage is resisted, the others can still work. How do we know that this will work? Well, scientists have tested it out on our original fiend, Erwinia amylivora, using phages named Fifi044 and Fifi318. The results were promising, showing that phage treatment could suppress the pathogen and reduce disease load in apple plantlets. So, are phages the future? Well, one apple plantlet isn't an entire orchard. Outside, plants experience changing environmental conditions which could affect phage activity, and equally, we need to consider how we could industrialise phage production. So, it's still a maybe. Phages probably aren't about entirely replacing chemical treatments just yet. Instead, they could give us a much more targeted tool to control bacterial disease, protect agriculture and crop supply.